Welcome to Neurosalience, the OHBM podcast.
Welcome to the Organization for Human Brain Mapping Neurosalience podcast. I'm Peter Banatini, and here I discuss all things brain imaging with scientists from around the world. Today, my guest is Daniele Marinazzo, a full professor in the Department of Data Analysis at the University of Ghent in Belgium. For over a decade, he has been showing us what further information and insight we may extract from brain imaging data from EEG and MEG to fMRI. He's technically a statistical physicist, but in reality, he's a network neuroscientist and a data modeler who is constantly pushing the envelope. In this podcast, we discuss some recent papers that go into how we might be able to improve the impact and relevance of new findings and models through careful benchmarking and well-considered experimental design. We talk about his desire to move from correlation to causation in functional connectivity studies, as well as moving from pairwise correlation analysis to multivariate correlation analysis. We discuss granger causality a bit and also delve into the limits of hemodynamics, limits that may be pushed back to a degree as suggested by his compelling work that shows that the hemodynamic response function, which varies over space, may be estimated on a voxel-wise basis using resting state data alone. We then delve into his work in estimating and mapping the excitation-inhibition ratio in the brain using gamma frequency coherence as a signature. This has potentially profound impact, both clinically and in research. Lastly, we discuss his collaborative work with the European Human Brain Project towards the creation of the useful website called eBrains that serves as a repository and tool for exploring shared data as well as code, and also providing a user-friendly encapsulation of the project's collective effort. This was a fun, eye-opening discussion with an outstanding scientist who is not only deep in the trenches of network modeling, but also a strong proponent of open science and constant engagement across disciplines. I hope you enjoy it. Welcome, Daniele.
Welcome, Peter. Thanks for having me.
Yeah. I usually start out the podcast with a little bit of history. I've known you over the years. I was just mentioning to you before the podcast, I remember inviting you to the editorial board of NeuroImage. And I think that your influence has been significant. I think there's a number of people in the field who are sort of these people who do very rigorous and careful modeling and actually try to remind the field of what they can do with the data. So, but you had actually, just to start out though, you had a very interesting, sort of history in terms of how you got here. It's a lot of, everyone gets to where they are in different sort of ways. And, you started out as a physicist and you got your master's in physics, but then, just tell us how your path from there to where you are now.
So, even if sometimes I describe myself as a statistical physicist, I sort of became like that. Let's say that a statistical physicist is someone that doesn't understand about MRI physics, doesn't understand about astrophysics, doesn't understand about quantum physics, doesn't understand about string theory and all these things. My minor was in solid state physics, and I decided even to do my master thesis in the chemistry department because it was the fastest way out of the faculty. I wanted just to finish and get over it. Then during my master thesis, which was on the other hand on Monte Carlo modeling of ionized gases with Fortran, I became passionate with modeling and with the research process, if you want. And motivated enough to start sending application for PhD position abroad, and eventually I got a PhD position in the Netherlands to work on laser physics. So the idea was to accelerate a gas, so ionize a gas, making waves, and then have electrons surf these waves in order to accelerate. That was a nice idea on a tabletop setting. For one reason or another, I ended up a bit hating it, and would probably have stayed there and then say, well, okay, let's be resilient, let's go through it and so. But then, let's say, a minor illness took me away from the lab for a couple of months, and so I had the chance to say, no, well, actually, it's not what I want to do. So I started a research position in a newly opened center at the University of Bari, where I had my master's degree. So the newly opened center was signal processing in a wide sense. So there was some biology, some physics, some chemistry, some medicine, etc. And then eventually I started my PhD. The PhD was on the analysis of biological data in general. So I started with DNA, cardiorespiratory analysis, and so. Then, during my PhD, I was suggested to go abroad to learn a bit more about machine learning. It was the old style machine learning, so the kernel functions, all these things. And then I decided to go to the Netherlands, to the where Bert Kappen was. Unfortunately, for miscommunication, Bert was going in a sabbatical exactly at the same time. But Stan Gillen, the head of the department there, also decided, well, I'll have you anyway. We can do some computational neuroscience. And then everything a bit started, because I met some people there who got me into the first SFN conference with some project on computational neuroscience and so on. So eventually, I decided to stay in computational neuroscience and even to do an experimental postdoc in Paris in vivo electrophysiology, even though I was still a bit scared after the experience with my first PhD of experimental work. Not sure if it worked out. So, but I mean, yeah. In the Netherlands, the first time I broke an expensive rod of material. So, and of course, the first rat that I tried to anesthetize in Paris, it died on the spot. But anyway, it happened. But eventually I got deeply, let's say, into neuroscience and then computational neuroscience. So, and then, when the time came to look for a position, there was this position in Ghent, and among a few others. So actually, I got the luxury to choose between a few of them, and I know this is a quite. I feel lucky about that, and I also feel lucky that I had the possibility of choosing or so, and also that I had a very enthusiastic and supporting partner, which is also important. So we were in Paris, and they say, "Well, Ghent, I don't know where it is," and she said, "No." The Emperor Charles V was living there. It cannot be that bad. So,
yeah. Yeah, well, that's actually okay. Yeah, that's so. So they're right. They have so it's key actually, right? And going any place, right? You consider the location, the lifestyle, the quality of life, but also the academic environment and all these factors. But it's also interesting that you mentioned dabbling in experimental work, and that's one thing. Just as a short aside, is that I'm realizing that more people in the field of neuroimaging have never done an experiment. Most of them are sort of just analyzing data, and I wonder. I think there's always value in sort of like getting a visceral sense of the data, just even a little bit, just doing some experiments, just to you have. I think it guides one's thinking when grappling, when just trying to, understand artifacts and noise and confounds and things like that.
Yeah, I mean, it's definitely an added value, which I would recommend. Then, let's say the analysis and modeling part is, let's say, diverse enough that, let's say, one can have can stay there forever and satisfying the curiosity and so. And I have to say that sometimes the same feeling of I don't know, getting your hands wet. Sometimes you can also have it with data. Then it depends. But I'm definitely glad. I was exposed to different type of data acquisition. So the real, let's say, surgery and electrophysiology on in vivo on animals and so, and then also other type of experiment like EEG or fMRI, of course.
That's great. That's great. So as far as what you do now, so before we get into some of the discussion of some of the papers and some specifics of your work, so, and you sort of alluded to it and I've already talked about it. So you would call yourself right now, you're a statistical physicist, but you do most of your work is involved with aspects of network modeling. And of course, in the field of neuroimaging, there's many types of models. I mean, there's models all the way from modeling, action potentials all the way up to, network modeling and beyond. So as far as what you do and what is, just to build a perspective before we start, the value of, of modeling. I always try to tell people that, when they, fMRI is sort of like, oh, you have a brain activation and it's a certain place, but it means nothing unless it's sort of tied to a model and either a hypothesis or something. And it seems like it's important. And so what would you say you do and the significance of the area of what you study in?
Yeah, so the easy answer is that if you stretch the terms enough, network modeling could be basically the analysis of everything that is out there. Because a network can be seen as really a pathway, so different places connected by a physical or anyway a link with a cost. And sometimes people doing network analysis forget about this aspect of network. But on the other hand, a network can also be seen as an expression of collective behavior in a wider sense.
Yeah.
And in this sense, all the systems worth studying display some collective activity of its components. So these are for the network. And for the modeling, of course, can be a very specific model with a lot of parameter constraints and so. But indeed, whenever we get to analyze some data with the correlation, with even with the probability distribution in our mind. So when we are surprised about seeing some data or when we're not, it's all about modeling. So yeah, the easy answer is that the network modeling is basically everything. Then more specifically, of course, we can say that different type of, the models reflect our assumption when we analyze our data. So the idea is that when we design an experiment, even before having data, so we have something in mind, and then we work on this and we try to interpret this. So maybe we'll get to it later when we talk about what we can learn about from correlation or functional connectivity, etc. Yeah. So, but and in the same way, even if I don't always use Bayesian statistics or so, we are also all Bayesian if you want, because we have some priors and we change it or so. So it's
yeah.
Anyway, I don't want to be over generalistic or so, but let's say let's say there's definitely value in modeling and in seeing what we study as networks systems in general. So as a behavior of some units which behave in that way because they are together.
Yeah. Actually, what you say sort of reminds me a little bit of way back in one of my first podcasts. I was interviewing Danny Bassett, and I kind of came to that same conclusion. I said, well, literally everything can be modeled as networks, and she sort of thought about it and said, well, there's also fluid modeling and And actually, that's interesting. I mean, you could actually, some people have done fluid, modeling of the brain as well. And
yeah.
So, but it's a slightly different, framework. But once again, you could probably even they can converge in some way too, but still.
Yeah, sure.
Yeah. So anyway, so going on to, it's interesting when I asked you if there's any papers you thought you'd want to talk about. Usually with my guests, they send me, and you did, and you send me their papers, but I was intrigued by the fact that you sent me someone else's paper, and I actually had not seen it before, and so it was eye-opening. This really nice paper by Mika Rubinoff in eLife, and just published in 2023, Circular and Unified Analysis in Network Neuroscience. And the more I read it, the more it did have that feeling of, when the paper on circular analysis and data analysis came out, it was sort of an eye-opening sort of wake-up call to the field. But here, it was sort of a wake-up call, it seems, to, and I'm still trying to get my head around it fully, but it seemed like it really kind of was tough on, a lot of the work that essentially, as I try to encapsulate it, everyone wants the field to move forward. And he feels that there's all these speculative models and only a few benchmarks, and none of them actually have traction because they're always comparing themselves to other speculative models, and not really making that much traction. And he really, goes into everything, including, talking about oscillations, talking about internal representations and things like that. All those are sort of speculative models that are there and they're interesting, but there's nothing, they lack traction because they're not, put up against some sort of ground truth that's been established. So what's your take, and why did you suggest this to me? That's just my take on it.
No. I mean, it's a paper I love, and I love the idea. Then there are also some parts when I read it that I maybe find it even over specific, in the sense that I am afraid that someone who reads it not with a, let's say, with a critical or with a sobering mindset can think, well, okay, no, but I don't do this or whatever. So it's this is too specific. But indeed, right. We sort of made a similar point, for example, in a paper that maybe we'll discuss later about going from association to causation with functional connectivity. So it's an idea of having, let's say, the map and the territory. So the level of the target properties that we want to observe, and then the level of the observables, because sometimes we cannot observe the actual things and so, and then the level of inference. And sometimes we get lost in this mapping. Or, for example, we try to model our system with a certain mathematical tools, and then we end up giving to that system the property of the mathematical tool. So I don't know, the critical brain. I know I make only an example, right? But it's I mean, I don't want to go even into the epistemology of science, because it's really not about is not what I do, and I don't want to step on other people's toes. But even more crudely, it's just sometimes getting lost between where we want to go and the tools that we have to observe it. So, and this, in this sense, was the circular part Mika was talking about. So, let's say, validating against something that it's already in our mind, which on one hand is good because we want to have an idea of the phenomenon that we are observing. But then the same tool that we use is basically meant to validate that same thing. So.
Yeah, and actually, that you bring up a good point. I mean, you're right. I mean, I think that some people in the field might push back a little bit and saying, "Well, this is sort of like we're in this exploratory phase, and we're coming up with all these tools, and we're sort of trying to interpret it." And basically, what he's saying is, it's just it's good science to, for instance, and maybe this is a slightly different point, but still along the same lines, is like when you mentioned criticality. There's actually somebody here at the NIH that Dietmar Plenz, who's very
Yeah.
much into that, and I'm really fascinated by that. And so and that's what I think that's what happens is that people who enjoy these concepts are like suddenly they think, oh my gosh, the brain is a critical system and it's optimizing itself to transfer information, but we don't know. So that's the thing. It's like it might show properties of criticality, but we don't know what it's for. We don't know what it's doing it for. It could be doing it that way, and we don't know how it's doing it. And so there's so many unknowns. And I think it's important to hold back on over-interpreting what our models say before we fit it into a wider landscape. I think that's my take. And even things like oscillations. A lot of work is being done on oscillations in terms of saying, okay, well, there's these oscillations. It's for coordinating. It's for doing things like this, and it might be, but there's nothing that demonstrates that conclusively. It's just sort of a phenomenon that we're observing. I think that's what it was trying to say overall. I mean, those types of things, and I love the idea though of, and also vision scientists. They talk about internal representations all the time, and it's and you wonder. I mean, it's basically these are sort of like tools so that we can think about what they are, and there's this balance between a model, a strict model of like, okay, that's what it actually is, versus kind of what we use, the words we use to talk about it and think about it. And so it's a really slippery slope to whether you fall into this sort of wider belief system about this. But yeah, no, I think actually it's definitely worth reading. He came across pretty tough on a few pieces of work, and I did sense, when I started reading, I'm like, yes, this is what the field needs. When I go to neuroscience, I mean, this is sort of a little bit sort of tangentially related as well, but when I go to SFN, Society for Neuroscience in the U.S., it's overwhelming, and you have like 30,000 people all doing this research. It would be great, and they're all showing something, and you're overwhelmed, and it's like you get a lot of ideas, but do you really feel like the field is ratcheting forward? And I think this sort of speaks to that, where it would be nice to have some unified model that everyone sort of agrees upon. And then if you have new data, you either fit it in or change the model, or it has to do something, or else you discard it. I think that he's trying to say that we need a process like that. It's interesting. So anyway.
Yeah, no, definitely. I mean, then again, in the quest of a model which is general enough, that again there is the possibility of getting lost, or anyone seeing something in the elephant, so the trunk or whatever. So it's
yeah.
But anyway, I think it's good to think about those things, and to ourselves to see, okay, where am I in this moment in going from the level of observation to the level of inference, etc. So
yes, yeah, and I think right. All it comes down to is sort of a reminder that we have to be careful and about what we know and what we don't know, and what we're speculating. Yeah, and so it's good read. And but anyway, let's get on to a few other things here. Before I get into, and actually it does sort of tie a little bit more into your work on connectivity and inferring causality. So and then I'll jump back on excitation and inhibition, because I was really fascinated with that piece of work that you're doing, because I think that struck me as really cool, like what your observation is. But let's first go to causality and functional connectivity. And I was sort of reading a few of the papers that you had. So you had this Nature Neuroscience paper in 2019 that you co-authored with a lot of really significant figures in the field, advancing functional activity research from association to causation. And as I'm reading this, I did like your analogy that you had at the beginning, looking at describing the moon. It's like, well, you could describe the moon as the photons just hit your eyes and just say, okay, well, it's sort of correlated with that. Or else you could try to say, well, the moon is this, and describe the physics that describe it. And so that's sort of what it's trying to get at. I'm trying to understand, and as I was reading the paper, I was trying to get a practical sense of all the things that limit me from, like, when I tell people, oh, you can't say that's causal because you're making too many assumptions.
Mm-hmm.
How do you, and so basically you're saying, well, you can get beyond that. And so what is the message of that paper to go from saying, okay, well, you have functional connectivity, you have these networks, they're correlated with behavior, and people can say, well, there's a correlation, but how do you, and yes, you have experimental manipulation that could change that, and then the behavior changes, the network changes, but still it's sort of a associative. How do you get to saying, well, that is a cause? And most people point to like using TMS or something to disrupt the network, but here you're saying something else. So
yeah, I mean, sometimes we can do that, and we can have our modulation, which is a real, I don't know, silencing or enhancing of some mechanism that we know. But sometimes even a modulation which is not a clear intervention or lesion or so, if properly thought of, can help to answer a question. Then the idea, and it recursively comes back in discussion, is, okay, is it worth to ask questions about the brain even if we don't get to the cause, or are the questions all causal or not, or so? So even in, I don't know, in the classical sentence, correlation is not causation. Yes and no, because ideally, I mean, we know what a correlation is, and we use it widely. So why? So unless we admit that we just go and appear and apply Pearson correlation to random stuff and see what comes out, the idea is that when we correlate two variables, we have a question in mind. And this question, hopefully, is a causal question. It's a causal question about the system that we want to study. And so if we have the correct experiment and we know the tools enough, then we can definitely give an answer to these questions. And to the extent that the questions are causal, the answer will be. Then someone will say, okay, but you will never be able, especially in large-scale human neuroimaging, to design the perfect experiment because you have too many confounders. And this is true. You cannot go to any single part of the mechanism. But this goes to another distinction that sometimes we try to do, especially when we work with this type of systems, between the mechanism and the behavior. So what we can observe, what the system does, and how the system is made. Of course, you cannot separate the two things, but we have the observational level, which sometimes is the only thing we have, and this is what started the scientific process. So observing things and then asking questions from our observation since the ancient times. So this is a precious place to be. We don't have to feel bad about observing a behavior. And then we have something about characterizing this behavior with our knowledge and with all the inference tools that we have, and also, of course, with some humility and good faith, because we also need this. But let's say the idea of the paper is that, okay, we can navigate these levels between what we cannot observe, what we can observe, and what we can infer with a clear mindset. And also, sometimes, I mean, the paper was sometimes spread by the fact, okay, but it is association and causation, and then also about granger causality, which has an unfortunate name, or the causal language in general. But then if you think about it, the causal language is not just using granger causality.
Right.
Causal language is everywhere. When we say, even in this super nice and very thought of experiment by the Dosenbach lab, you had the Evan Gordon there. So, and then you say, okay, this newly discovered network encodes this and that. This is causal language, and they use correlation there.
Yes.
And it's a good thing.
Yeah.
But it's more widespread than simply talking about the C word, let's say, the C in the sense of cause. Yeah.
Yeah. So I mean, right? You could. So yeah. So I think that. So once again, it's about model building in some sense. If you have a series of experiments that are associative, and then you sort of logically parse the experiments in such a way that you can actually then sort of dig down and then have a stronger inference about causality just by the careful modulation of the experimentation. That's essentially what I think that. So there's sort of a continuum. It's not like it's associative and we can't say anything about causality, but then the more different experiments you do and the more different results you get, the more you can then infer causality, it seems. In that sense.
Then the other question can be, why then are we so fond of Pearson correlation while we have many other better methods, starting with granger causality, which is already a huge improvement with respect to correlation. And this is, when I started a bit in the field, and when let's say my colleagues and supervisor back during my PhD said, okay, why don't we study this granger causality and also some nonlinear extension of it? So, I mean, I find it somehow surprising that it was not used. I mean, if we can condition about something and this makes better inference about the system we are observing, then we should just use it.
No, I think actually. So let's just talk briefly about granger causality. I think that a lot of people in the field, it's obviously a general tool that can be used in many different ways, and it seems like. And so am I correct in saying that the general implication is that you're relying on delays to infer some level of causality, and certainly that's true with MEG or EEG or some electrophysiology where there's hardly any. I mean, there's noise, but there's not systematic delays due to non-neuronal factors potentially. I think where people now cringe. They don't really cringe. They all the fMRI people who are the physicists, including myself, we all know that, and you talk about this, and this is actually good when you talk about correlation, your methods, and we'll talk about this too, and that is that in the brain, at least when you're using hemodynamics, anywhere in the brain, depending on the vessels, it's plus or minus. Some people would argue plus or minus like 2 seconds potentially. So if you try to just apply granger causality, focusing on delays there, you're going to be kind of lost in a sense that it's sort of lost in the hemodynamic variability. Mm-hmm. But I don't think that, there's many other ways of applying granger causality that are outside of that context. And I think that's worth clarifying. So would you agree with that? I mean,
But I mean, again, the features of the hemodynamics and the nature of the bolt signal has some problems which are definitely not specific to granger causality.
Yes.
So the hemodynamic, even considering one single time series, complicates our view of the neural system, which is beyond. So when what we get to observe is that thing. So it's like you want to study something in microgravity, but then you go out there and there is the gravity, which is the hemodynamics and all the physiology and so on, which changes things sometimes in an irreparable way. So we don't even have the luxury of saying we remove the hemodynamics and we get to the neural part, even though, I mean, definitely, I've been guilty of that in the past maybe when the convolution work was sent out in 2013 or so, saying,
Yeah.
okay, we deconvolve the hemodynamics and we get with the neural part. I mean, it was an assumption that we
Yeah.
have some neural events but again, so this is not a problem which is in general for granger causality. Then, if you apply granger causality, there are some specific work which has addressed this. So, for example, starting from there was a comments and controversies issue of Neuroimage in 2009 on connectivity or so, where there were different papers or so. There was a very nice paper called Affective Connectivity, Influence, Causality, and Biophysical Modeling by Pedro Valdesosa, Alan Rubrock, and so, who really tell what we can expect from causality measures in neuroscience and what the role of the hemodynamics is. And so it was back in time that there was this study by Freestone and Olivier David on the influences in a rat model which had a weird and very long hemodynamic response, and so, which of course messed up with the delay. So there are some things that can be addressed. And then the group of Daniel Seth also did some very nice work on granger causality of fMRI bolt signal is invariant to hemodynamic convolution but not downsampling. So saying that the problem is not the hemodynamic per se, but the fact that the hemodynamic is also downsampled.
Okay.
And then another one called behavioral granger causality under filtering, theoretical invariance and practical application, which again, it's a part due to the low pass filter, but part due to the downsampling. Okay. And then a recent follow-up with Leo Novelli and Ada Razi, again with Barnett and Seth, which is called minimum phase property of the hemodynamic response function and implication for granger causality in fMRI. So those things have been studied. And sometimes they are sobering, and sometimes they say, okay, we can be pretty confident. Then, for example, I don't know, even my blind deconvolution approach, I started it with the idea of deconvolution and having better granger causality, which at the moment I was happy with, and then I ended up with the other part. So I wanted to throw away the bathwater without the baby, and then but then I stuck with the bathwater, because then in the later studies I found it more sometimes interesting to look at the variability of the HRF in different parts of the brain and so. But again,
just to focus on that paper for a second, I really like that paper. And actually, you pointed out, I skimmed it at first, and I thought, oh, you're just doing with one human. You're actually from resting state. You're estimating the hemodynamic response on a voxel-wise basis or a region-wise basis, and just by looking for any deviation, and then doing some sort of modeling of that around that time window, and sort of going through, and that that's and definitely you can probably characterize the hemodynamic response of the brain, but still that only gets you, probably because of the sampling rate, it still only gets you about to a second or two certainty, which is still maybe not enough to infer neural causality, but still it's interesting. I mean, in itself, characterizing hemodynamic response delays in the brain, throughout the brain, I mean, that's could be useful clinically. It could be useful, and also it's useful, as you mentioned, for just simply calculating correlation. Because many times I remind people that if you just use one set latency, when you're doing correlation with anything, you're always assuming that everything's lined up temporally, but it's not necessarily. So it was really a nice paper. But also interesting to me is if you could do that robustly, and I assume that you're pretty close to it. There's been other work by Cesar Ribeiro Caudas in Spain, trying to do deconvolution to get at events. And it seems like you could, it's the same type of thing where you could actually then.
So I would say they are similar. Let's say our approach is even more really top down. So you only need the bold. On the other hand, the work by Cesar, which I greatly appreciate also, and similarly also some work by the group of Dimitri van de Ville. So starting with the total activation with Ishik Karanoglu back in time when this paper also came out. It's a step further versus having a generative model of the process. So, for example, of distinguishing between innovation and so the activation as innovation, so which is very elegant and goes, if you want, even closer to the mechanism, if you want. So, in this case, it's let's say a really top-down approach based on the fact that on the point process theory, so you have some event that generates a peak. So, for example, this approach would not work in a breath hold paradigm, because there you're assuming by default that you don't have a hemodynamic response elicited by a neural event, let's say.
Right. So you just that's the one assumption, and certainly there's a lot that could cause, event, fluctuations like breathing and heart rate and motion and things like that.
Indeed, we also have a paper saying how this shape is also modeled by, not directly by the physiology, because the physiology, unfortunately, in fMRI is inexplicable from the final product, but to different processing techniques which deal with different aspects of physiology. So different types of regressor, removing some nuisance, etc. So
I think okay. So this is actually and I really do think, there are techniques out there, but I think this is something that everyone should be using. It came out in 2013, and I'm surprised that. Sure, there's a little bit of computational overhead, but you could actually just apply this to all of your resting state data, and I think it would clean things up a lot. It would also extract interesting information that could be useful too. But
we did let's say all the effort to put it. So, for example, it's also included in Brain Life, which is this excellent platform by. Initiated by Franco Pasilli and colleagues, and so in which you can just plug in and cloud compute using your bits data, everything. Yes. So, and then is also on eBrains, which we will maybe discuss later or so. So again, it's it's indeed they're ready to be used and to be possibly expanded. So because indeed also when we do large-scale modeling of computational, of a large-scale brain activity. You model the local field potential, and then if you want to compare it with fMRI data, you convolve it or you use, for example, the balloon wing castle model. Yeah. And we have shown in a limited data set, but one, for example, in which the hemodynamic could indeed change because it was a brain tumor data set, that instead of using the balloon wing castle model plugged in after the local field potential, if you retrieve the hemodynamic response function from the resting state data and then you convolve this one with your local field potential, then you have a better similarity between the large scale pattern that you have.
That's really nice. That's really, that's an important finding, because that's, I think, right. It's something that people should be aware that you can do, right? And it sort of gets at the hemodynamics directly, and as opposed to just using this model up front. Yeah, that's great. And of course, tumors are an extreme example, but the hemodynamic response does vary throughout the brain, and it's not, and it still hasn't really quite been established how it does. And it would be great to make a map of that for every single subject, and at least you're getting closer to that here, and I like it. It's really. And have you tried? And the first thing I thought of, and I'm sort of just catching up with this literature for some reason. It's funny. I try to follow literature, especially along these lines, but I missed this a little bit. But have you tried just comparing empirically? Like, you do this estimation, and of course you get those estimated response, let's say in the visual cortex. And have you tried doing an event-related visual response, task, where you then compare empirically the event-related response to the derived response?
We did two things. One, in the visual system was, let's say, typically you compare, let's say, the the bold signal with the envelope, for example, of the alpha signal in the in electrodes close to the visual cortex. So typically in the occipital electrodes and so. Then we saw that, for example, if you deconvolve the HRF, and then you use, instead of the envelope of the EEG, so you take the EEG and you convolve it with this new with the HRF, then you have a better view of this modulation and also of the relationship and the inversion of relationship, for example, with the thalamus between thalamus and visual system. So the inversion of the height of the HRF in different states of the awareness. And then we compared the spatial maps of the resting state HRF with the task HRF, and also we found a correlation at the level of the spatial maps.
That's great. That's great. That's really cool. I really like it, and it might be able to draw further inferences about what drives even resting states. So people need to realize that it's probably a series of events that occur as opposed to some sort of oscillation or continuous activity. And this sort of infers that. We're doing other time series analysis in our group, edge analysis and things like that, which is sort of just another way of looking at, trying to pull out these events and correlated events. But yeah, no, this is great. And there's so much more work that can be done.
Another way, if you want the edge analysis, if you talk about, for example, this new approach by the Sporns lab, so it's basically the same idea of these bold activations that let's say Dan Tagliavo and colleagues first observed. In their case, they were relating it with the critical behavior. So, but again, they called it the R beta. So the part of the bold signal after a certain threshold. So these events were, let's say, enough to explain a big part of the variability, which was then going, let's say, which was revisited also in what was called the co-activation patterns by Xiao Liu and Jeff Dine, and then all a line of research there. But let's say it's basically the same thing. These peaks. So then, of course, these peaks are a sign of variability and not necessarily a sign of non-stationarity. So because even for Gaussian signal, you will see that the extreme values are more correlated. So, but again, this is about navigating the observation and interpretation.
Yeah. So that's actually a great example, right? That ties that in. So now this is there's these other approaches that are converging, and it's a rich area of work. And I think, I think that rather than, it's certainly useful for looking at correlation, looking at neural activity and inferring that, but it's also, I really do think that there's interesting signs of vascular dementia or things like that might come out of this sort of way of looking at the data that could be useful. Yeah. So, okay. So great. Now I'm going to jump back to your really interesting work on looking at and trying to assess excitation and inhibition balance. So, this is a little bit more outside my area of expertise, but the idea is that TMS or other things can modulate this. It's maybe it's one of those things that I think is maybe looking at susceptibility to epilepsy or things like that, maybe. But so, it seems that there's the only way in the past of trying to get at this is just by stimulating and looking at the responsivity of an area, stimulating with beta bursts, and but it seems that you have a couple of nice papers that talk about using, a number of techniques, but using, like, for instance, MEG or EEG phase synchronization, and particularly at gamma frequencies, as a measure for excitation inhibition balance.
Yeah.
And so you can map that throughout the whole brain. So could you talk about that a little bit?
Yeah, definitely. So let's say excitation inhibition is, again, one of those things that if you stretch it enough, it equates with brain function. Because, of course, without excitation inhibition balance, then the brain doesn't work. And then, from the point of view of a physicist, so even the simplest, one of the simplest models in physics, which is the Ising model for magnetization, shows interesting behavior in the sense of long-range correlation and critical behavior. So at the critical temperature. So when the magnetization goes the system goes from chaotic to frozen, and then there is a part in which interesting things happen. Then, as a matter of fact, if you try to simulate large-scale brain activity, putting Ising spins on the structural connectome, you get very good fit, similar to the one of biologically inspired models. So, which, again, depending on the model that you use, you can call the critical regime, you can call it the balanced state, if you want.
Yeah.
Or if you use the Kuramoto model, which is a model specific for oscillator, again, it's a balance between how an oscillator is reactive to the others, but also independent. So it's always a thing about balance, not to mention the fact that, indeed, without balance, either it's dead or it's extreme epilepsy, then dead.
Yeah, so, exactly. Yeah.
Now, and this is, again, something in which we get lost between the target properties and the names, the map and the territory. So, specifically to this, and for example, one of the things which has also been associated to excitator inhibition ratio is the aperiodic part of the spectrum. Now there is I don't know if you're following the EEG literature or in the electrophysiology. So there was a paper and then a toolbox from the Wojtek lab who did excellent work in putting this to the world. Something that, again, had been observed before, in the sense that there are even pure oscillations that wax and wane can give rise to something which is not which does not contribute to spectral peaks, but to the spectral slope, so the aperiodic part of the spectrum.
Okay.
On the other hand, this has been called as the dark matter of the brain or things like that, which of course is not. But again, it has been related with excitation inhibition ratio, of course. Is a bit like when you see the DMN is correlated with this and that. It's very sensitive but deeply unspecific. Now, I had the luck during my sabbatical post during COVID. I decided to visit a neurorehabilitation hospital in Venice. And it was extremely enriching because I met some people there doing excellent work, both in neurophysiology and in fMRI. Yeah. And in particular, this colleague, Giovanni Pellegrino, who is a clinical doctor, is an epileptologist, and so he really knows and really cares about excitability. And so whenever he reads on the paper, okay, there is this property extracted from the phase coordination, which we call excitability, then it comes to me and say, Daniele, can we call this excitability? And I mean, sometimes I try to play the devil advocate and so, but again, we try to see which of these measures can, let's say, actually measure excitability, because Giovanni and his colleagues had a very well thought experiment for theta burst simulation or epileptic subjects and so on. So, for example, we tried different measures, and indeed one that correlated reasonably well with the actual excitability was this excitability index as defined by Meisel et al. In PNAS 2015 paper, which is basically the equivalent of the intertrial phase coherence that you use across trials to see, for example, if a stimulus resets the phase. Because if you do ERPs, you know that, the idea is that if you sum and average different things which are not time locked to a stimulus, then the noise disappears, and so you have the event-related potential. But if the effect is not on the phase, then the different phases can zero the overall. So you can have a prominent phase effect, which does not show up in an event-related amplitude, let's say.
Okay.
And so to do so, it's used the so-called intertrial phase coherence, which is basically you make if you want, an histogram of the phase at each signal and see if it's narrow or not.
Yeah.
On the other hand, the Meisel et al., they did it especially. So in different locations of the brain, different vertices, they found this phase consistency, and they propose it as a marker of excitability. And so with some experiments aimed to elicit excitability, so theta burst, or for example, steady state auditory response, which is again something which very powerfully makes a 40 hertz wave in the brain.
Yeah.
And not only in the auditory cortex, but it spreads. And then it's also not clear whether it spreads like throwing a stone in a pond, or it spreads with different other modalities. But indeed, we saw that this measure had promise in actually getting to excitability. Other, less. It's true that is by no means a complete or final word, because especially when you do this type of work, invasive work, or so, you don't have a huge sample size or so. But again, it's a certain point, and I consider myself very lucky working with some people who really know what excitability is, and they care about it in a clinical context.
Yeah, no, it's so okay. So just so I understand, so the mechanism, and there's probably, it's not clear exactly what the mechanism would be, but my intuition sort of tells me that like the phase coherence suggests that if you have, causing neurons to fire, it's like if you have more neurons in coherence, they can sort of add up constructively to sort of cause firing. And that's sort of my intuition, but that's still a little bit different than what you're taught what one is taught is in school about excitability, is that the membrane potential of each neuron sort of goes closer to threshold. So there's two different ways of sort of thinking about this.
Yeah, indeed. I mean, and there again, I don't want to delve neither into the deep biology because I don't know, nor in the scale because this is with a large scale. So, and I don't even want to delve into philosophy, but the name excitability is again something that, in terms of Aristotle, would be actionality versus potentiality. So there is excitability. It means that already, it has the potential of being excitable. On the other hand, you are measuring something that happened already.
Yep.
Sorry, Aristotle. I always bash on Aristotle because then I say that the granger causality doesn't get to Aristotelic code. But in this case, Aristotle is safe. Let's say. He's right.
Yeah. The imprecision of words in that regard. But and actually, you mentioned clinical applications. One I saw, I just happened to see in iScience here was white noise. So white noise just seems like it reduces excitability in most of the brain, except in one area along the superior temporal gyrus or something. It was fascinating. The idea that you could potentially use that as therapy for epilepsy or something like that. It was very cool.
Yeah, and also, for example, when you are in the scanner, sometimes even the scanner noise can change the level of excitability or so. And on the other hand, indeed, noise can sometimes facilitate some effect, even synchronization. So there is a phenomenon in physics called stochastic resonance, by which actually some synchronization between different units is facilitated by the presence of noise.
Okay. Yeah. So it's okay. All right. Well, not to switch gears too quickly, but I really wanted to talk a little bit. So I think we might. You also had recently talked a little bit about. You had a nice paper, and we on looking at, instead of pairwise correlations, going beyond pairwise correlations.
Yeah.
And, is this something that? Is this sort of like a blind spot in the field that we're missing? we're not looking at
Well,
nonlinearity.
apart from being my obsession at the moment, again, it's something that if you think about it, if you look at the system as a network, why should network be a pairwise one?
Yeah.
On one hand, we don't have to make things unnecessarily complicated. On the other hand, if we like the idea or that the brain works in parts communicating with each other, and we go to a network representation, but network in the wide sense, not network in really sense of links and nodes, which can also be, there are the hypergraphs for that, but
yeah,
why should the interactions only be dyadic?
Yeah.
And this is true both at the mechanism level and at the behavior level, because you can have, you definitely have parts of the brain linked simultaneously to different other parts of the brain.
Yes.
And also, even if you have a system which has purely dyadic mechanism, you can then have interesting higher order effects. So you see this in the easing system in the frustrated case, even with 3 spins, but you definitely see it elsewhere.
Yep.
And more importantly, things about emergence and also the trade-off between segregation and integration, which has been one of the paradigm of large-scale or even mesoscale neuroscience that we are looking at. Starting, for example, a typical example is the lab of Jerry Edelman in San Diego or so, and the two of his prominent scholars, Sporns and Tononi. Of course, Freestone there as well, but Carl's work, it's really, it definitely fits into this, but maybe he already figured it out before. On the other hand, let's say the work of Tononi goes into more the integrated information theory, which also had an interesting Twitter and offline theories, but also. And then Olaf Sporn's work on network neuroscience. So it's again a balance of segregation and integration towards function, and indeed a mathematical operationalization of the notion of synergy, of collective degrees of freedom shared by more than 2 variables, can be the key to understand these things. And this is important on at least 2 aspects. One is the purely statistical aspect. So statistical dependencies are biased, pairwise statistical dependencies are biased if there is shared information between 3 or more variables. In epidemiology, in psychometrics, everyone knows about the colliders, confounders. So on the other hand, in neuroscience, really little. So, for example, a paper by Mike Cole and Sanchez Romero from his lab was published into the Journal of Cognitive Neuroscience. So, a very, not a niche journal. Only a few years ago, where they were simply proposing to compare pairwise and partial correlation, just to see whether there is this kind of effect, which speaks to the fact that indeed there is a huge blind spot in the fields. And again, it's not just a mathematical quibble, some nerd stuff or wanting to propose something unnecessarily fancy. It's something really fundamental, which reveals interesting properties about the system that we are studying.
So how would one? So are there tools out there that? So most people would say, okay, I think it's easy to convince people that the way to do this.
Tools are getting there. So there are different types of tools with different assumptions. So this work in neuro was started a long time. Let's say 15 years ago by Stefano Panzeri and Simon Schultz on individual. So in talking about individual neurons and stuff, and so talking about this redundancy and synergy. So then some operationalization comes. So there was some Taylor-like expansion of the mutual information and similar things like that. And then of the transfer entropy we propose. All those things scale not so well. Then other things came. So, for example, the partial information decomposition by Williams and Beer, which allows to have redundancy and synergy as not two mutually exclusive quantities, but two coexisting positive quantities. And so, but in this sense, the Shannon theory of probability needs an extra condition. So you need to go into some postulates, etc. So already the theoretical field is a bit. There are hot but nice discussions about that.
Yeah.
A practical implementation is just the framework of condition probabilities. Then, if you want, and for example, for many of the data, it's better to relax the assumption of nonlinearity, because when one talks about entropy of mutual information, thinks about being a model-free assumption, which is never model-free, because also probability distribution is again a model. But even if you keep the framework of information theory, but you have a covariance-based estimator, which is exact for Gaussian data, but is probably good enough in many large-scale applications, because multivariate distribution tends to be more and more quasi-Gaussian when they increase of size.
Yeah.
It allows seeing the things and operationalizing in a way that scales much more graciously. So recently, Fernando Rosas, Pedro Mediano and colleagues developed a measure called O information, as for other information, which is again an expansion of the mutual information as a balance between the total correlation and the dual total correlation. So about shared degrees of freedom and collective constraints. And so it's if you want, about synergy or redundancy, about parts of the system that can tell together more things about their individuality, or cases in which you have redundancy, so the global information is smaller than the individual ones.
Okay. Well, that's interesting. That's even that parameter in itself would be, interesting to map. But yeah, okay. So
we apply this in a very simple case in fMRI, because in the global signal debate, the global signal has been seen as everything and nothing, pure noise, when on the other hand is the region of interest, is the whole brain. So it's not surprising that it contains relevant behavior activity, because it's actually the whole brain.
Yeah.
But it also contains all the confounders. And if you look at the correlation between the global signal and the bold in the vessels, it has a median of, for example, on the HPC project of 0.90 something.
Yeah. Yes. It's pretty.
On the other hand, they are two different things.
Yes.
And again, it's a baby and bathwater thing. So we were able, so partial information decomposition is specifically useful in these cases, wherein you have variables which look very correlated, but still they can have different contribution.
Yes.
And so we did this partial information decomposition between the global signal, the bold signal in the vessels, and the bold signal in different ROIs in the brain.
Okay.
And we saw that indeed the effect in terms of synergy and redundancy at different time scales is different. And then it disappears when you have simultaneous calcium and bold recordings. So you don't have this in the calcium, showing that it is really an actual effect carried by the bold signal.
Okay.
So this is very nice work by my former student Nigel Colombier, with again Lucina Udin, Russ Poldrack, and other colleagues.
Okay, I'll have to take a look at that, because that seems like
there is in your image maybe 3 or 4 years ago.
Okay.
4,
okay.
Yeah.
Okay. Yeah, I mean, it seems that's right. I mean, it's so tempting. I mean, I think a lot of the field is trying to confine people, based on platforms for analysis of locking them into sort of a standard pipeline all the way up to these levels of doing things. And I think that's premature in this case, definitely. Yeah. And I think there's, like you said, there's blind spots. There's implicit assumptions that we don't even know that we're making.
Exactly.
Yeah. That's really nice. That's a really good example. I'll definitely take a look at that. Recommend looking at that paper. And also at that work in general. I mean, certainly people have tried, and I just think about this off the top of my head. It's like, certainly if you take an area and calculate the correlation or whatever, you find, the correlation. But I think there's real power in doing partial correlations, and that's sort of maybe along those lines, saying, okay, there's elements of this that are more precise in terms of pulling this out. And that's sort of going the opposite direction of just looking at the correlation, but then taking multiple areas. I mean, you can go that other direction and.
Yeah, completely. I mean, but then partial correlation can be confounded because if you have two uncorrelated variables which influence a third one, if you do a partial correlation, these two variables will become negatively correlated, which is basically the very reason why, for example, in this paper with the trouble at rest by Ziad Saad and colleagues and so, it was the reason of the appearance of negative correlation in the after global signal regression. But and especially in the brain, because I mean, parcellating the brain is sort of artificial, because if you take different entities which by default are separated, there are no latent variables possible there, and then it makes sense to see whether they're correlated. On the other hand, sometimes you have, and especially in fMRI, you have a huge latent factor, which is the physiology, and not to mention things like the head movement, but even cognitive processes that recruit different brain areas at the same time. So it's artificial if you want to say, I take one region, which is, the anterior insula, and the other region, which is the dorsolateral prefrontal cortex. It's there is no separation between these two if you want.
Right. And so it's really risky to do that sort of thing. So I see your point.
You know by default that there will be some shared information. And shared information means interesting stuff, but it also means some trouble when you want to look at statistical dependencies there.
Yes. Yeah. Yeah. No, I completely agree. I think that, the field definitely needs more people reminding him of these things, these sort of things that sort of are clear to statisticians and or not even necessarily in the context of fMRI. It's not always clear. And certainly the people doing the neuroscience research are not fully aware of these nuances at all. So definitely needs more communication along these lines. Well, I just wanted to maybe this has been a really, really nice discussion overall. But I just wanted to end maybe a little bit with, a big part of, your website. You mentioned this, the e-brains, and it's mostly, as I understand it, it's, there's some equivalents in the United States, but I mean, at least this is in Europe. So what exactly is it, and how is it useful?
Europe financed huge project, even controversial, called the Human Brain Project, which was, a huge endeavor, which gave, in my opinion, very nice results. So I was only marginally involved in that, even if I have to say that the return of investment for me was very good, because I was there only for a few months, but in a period in which they were integrating the virtual brain, of which I was an early adopter with this large-scale simulator of brain activity, so into the Human Brain Project. And we did so with a clinical data set on brain tumor data set, and it was just before that the general data protection came into effect in Europe, making basically not impossible, but let's say tricky to share the data in a good way, because of course we can talk. But anyway, so our framework and our case study stayed in this knowledge graph of the Human Brain Project, which is basically a neuroinformatic platform collecting the results of the Human Brain Project for a long time, as the clear example. Now, one of the missions of the Human Brain Project was then to make this research available and usable outside of the consortium. And so eBrains constituted as a, let's say, non-profit organization to collect this heritage, and now it recently entered the roadmap for European services for research infrastructure, which contain different things ranging from, traffic, demographics, medicine, etc. So, and then there is eBrains, and basically the idea is that we need to make public and available, all the results of the Human Brain Project, and to empower people to run their simulation, to share their data. So let's say eBrains can be different things. Can be a platform where you can store your data or run your models, or it can be a research partner. So in terms, you apply for a grant, and then you list eBrains as a company which worked with you building some computational models that you are not able to do, maybe, or you don't have the capacity of doing so. Yeah. Or it can also be a service also to companies, the pharma companies. They want to test their models also. So it's let's say is a different. It has different faces also. And so when they launched it, I was contacted, among others, because I was being part of the Human Brain Project, and so our Flemish grant officers contacted us, and so the Belgian node was constituted. Now, just this, we are starting this eBrains 2.0 phase, which is a big infrastructure grant with more than 50 researchers from 19 European countries, and we will work on this platform. And I will lead a task which is basically is called Operate Digital Twins in the technical jargon. So it's basically about model validation and inference and producing executables in order to perform simulation, in particular clinical cases. And one of the good things that the Human Brain Project did in the last phase was to bridge the microscale model, so spiking neural models, with the large scale modeling of the virtual brain, and so.
Really? Okay.
So I will coordinate this validation and model inference part of this grant.
Oh, that's actually great.
Anyone can use it, even in the US. So you can become a user of eBrains.
Yeah, I might. Right. I mean, it seems like there's a lot of things. Right. I mean, it's interesting. You mentioned bridging scales. I mean, that seems like a fundamental.
Yeah. And one of the things that are missing there, and I suggest always to the eBrain people to do, but maybe not reinventing the wheel, but partnering with platforms such as, for example, the Brain Life or Open Neuro. I mean, with Open Neuro now it's easier for the bits and also for the so. But let's say my students or my colleagues from experimental psychology, which are less expert in modeling in this type of modeling, would still, in my opinion, find a too big gap in going towards the eBrains, because it lacks maybe online pre-processing part or so, which is also an important part. Yeah. That's why I also try to bridge this. And for example, I'm a mentor on behalf of the INCF, the International Informatics Coordination Facility, of the Google Summer of Code, which is a very good program because it allows students to work on a coding project. And at least in Europe, it's difficult really to hire someone as a technical engineer to work, especially in a small scale project. If it's a big scale, then maybe yes. But if you want, for example, I don't know, to make a toolbox interoperable and so either you are a nerd, or some of the people who work with you are very good at this, and sometimes they are, but sometimes it's not even a matter of being or not being good. It's just that either you do the science or you do the informatics part, and sometimes you cannot do both. So it's good to have this and to bridge the two things.
Oh, that's great. That's great. So right. I mean, it's hard in Europe just to contract people. I guess it's hard anywhere in some sense, because it's a highly skilled position to some degree.
Yeah. And the position, sometimes people prefer to take it in a company and not with a short-term contract in the university.
Yeah. Well, that's interesting. Okay. All right. Well, I will definitely dig more into eBrains. I was also intrigued, actually, on your website, you had a really interesting, the VOS viewer. That was also, I thought, I mean, there's a lot of tools out there. It's just a standard network sort of,
Yeah, it's very nice. Actually, too bad that you mentioned today, because just today I changed it with because I saw on Twitter automatic word cloud generator, and so I say, "Oh, let's try these new things." And now the BOS viewer is gone.
Okay, well, at least I caught it though, and you can still probably find it somewhere, I imagine. But
No, definitely, it's a very good tool also to do research, so not only to produce. Let's say net also because it was a pairwise network, so I don't like this anymore.
That's great. Okay, so just to maybe wrap this up, it's hard to wrap the whole thing up, but I think that there was a lot of information gathered. So is there any, and you definitely sort of answered some of these general questions as you went along, like blind spots in the field and what people should be doing or shouldn't be doing. So is there anything, considering, all of neuroimaging, what avenues, I mean, as far as giving recommendations to either young scientists or established scientists who are either skilled in processing or neuroscience, what avenues do you think, what areas, and I think we've implied this from the discussion, but what areas do you think are, have the most potential for making progress or deriving insight, or even clinical applications? What do you think is the richest area to sort of guide research?
Yeah. I mean, for me, and I'm sometimes really jealous of the place where I am, in the sense that I prefer to work on methods, and so maybe it's even I wouldn't say coward because it's too harsh, but let's say is a comfortable position, because then I don't have to go into then I have to run this experiment, I have to collect this data. So I'm extremely lucky to work with people who are extremely good in that and maybe appreciate the value of what I do. But let's say historically, I don't know, I see myself as a niche craftsman who does things that one day maybe we'll discover or not. But on the other hand, then I feel bad, because as a scientist, you also you have a mission to communicate, to improve things, and so on. And also the reason why I invest so much both in the scientific publishing, but also in neuroinformatics infrastructure like this one, is that at least there I can see that, and also in teaching as well, so I can make an impact on the
Yes.
For the rest, sometimes, for example, I don't know, I developed something for a very rigorous validation of statistical validation of higher order interaction. Then someone does it in a less validated way, and they say, crap. But on the other hand, it's also good that someone does it in a meaningful way or so, and with a clear application. So,
yeah,
in this sense, I would say that we should really talk more, and already between subfields. So sometimes I go to a physics conference and I realize that the network scientists and statistical physicists don't talk enough
right
to each other, and
right.
then not to mention the others. So I would say not to be afraid of this. For example, a stupid thing. I teach a course to the first year master student in experimental psychology, which until last year was called Analysis of Neuroimaging Data. This year, nothing changed in the topic of the course, but the title changed to Technical Foundation of Techniques for the Analysis of Data in Behavioral, and it's a mouthful.
Yeah.
But on the other hand, it's first of all to balance the expectation of the students, but also to see that we should really be able to talk more in a more holistic way, and get to know better both the tools but also the application. So I consider myself extremely lucky to have a work with colleagues who gave meaning to some things that I could have done. Some, I don't know, granger causality you can apply to climatology, you can apply to neural data or whatever. So it's
yeah, no, I completely agree with you. I think that people, it's an instinct. We get in our field and we get siloed by the terminology, and we have concepts that are just connected with that. And even within neuroimaging, even within processing, as you were mentioning, it's important to sort of try to keep those connections open, and try to, because we all get blind spots from that. And so, it's important to sort of keep a communication. That's something that should be emphasized all the more now. Without becoming too much of a generalist, know your field, but then know, you're right. Like, for instance, like you said, granger causality could be applied to many different things. And
so it's good to find a meaningful application. So sometimes even my students sometimes help me in this, because I teach to students in psychology, and they, with their questions, they help me greatly.
Yeah. I completely agree. All right. Well, so this is great. I mean, we could go on for hours, but this was really fun. And I actually learned a lot from not only preparing for this, but from talking with you right now. This is, and I get a sense that, there's parts of the field, before we fall into sort of a rut of a certain way of doing things, it's important to step back and read some of your papers, some of, Rubinov's papers and things like that to try to get a sense of, what we should do. So, all right. Well, thank you very much for coming on, and I look forward to seeing you possibly at the next OHBM meeting. So.
Definitely. I'm not sure I will make it to SAIL this time, but I really hope that we can meet each other soon, maybe at the next SFN or so. We'll see. But anyway, thanks a lot for having me. I hope the audience will like it, and thanks so much for doing this, because it's I know how hard it is, and this is really a great way of helping the field.
Oh, it's fun. All right. Well, thank you.
Thanks, Peter.
Neurosalience is brought to you by the Organisation for Human Brain Mapping. This week's episode was produced by Alfie Wine and Stefania Asimopoulos.